Hello and welcome to our last episode of the Happy Horse Nutrition Podcast for 2025. I can't believe it's the end of the year. As we wrap up the year, I wanted to reshare our most listened to guest interview from season 1, and it absolutely earned that title. This is my chat with the brilliant Dr. Belinda Chapman, who is quite possibly the coolest microbiologist you'll ever meet. And fair warning, once you listen to this, you will never look at your horse's manure the same way again. Because here's the thing. The microbes that live in your horse's hindgut and eventually end up in their manure don't just help with digestion. They influence pretty much every aspect of your horse's health, including how well your horse can extract energy from forage, how resilient their gut is under stress, how they behave, and how well their immune system functions. It is an entirely invisible ecosystem doing a mind-blowing number of crucial tasks. In this episode, we break down what the microbiome actually is. We meet the good guys that love fibre, talk about the weedy, greedy bacteria whose populations explode when the wrong food hits the hindgut, and we explore how fibre diversity in the diet supports a more diverse, more resilient microbial community and why this is important. Plus, there are some very satisfying analogies involving Swiss Army knives and food buffets. If you've already listened to this episode, biggest thanks, and I can guarantee if you listen to it again, you will definitely learn new things you missed the first time around because there is so much to take in. And if this is your first listen, enjoy. There is a lot to learn in this space and Dr. Belinda makes it fun to learn. Before we jump in, I want to wish you a wonderful end to 2025. I'm truly so grateful you've been here listening, and big thanks to you for helping us be one of the most shared podcasts of 2025. If you haven't already, we'd love you to hit subscribe so you don't miss any of the episodes coming up in 2026. All right, let's go immerse ourselves in the wonderful world of microbes with Dr. Belinda. Hello and welcome to the Happy Horse Podcast. Today we have with us Dr. Belinda, who is probably the coolest microbiologist you are ever going to meet, and we are going to be talking about the amazing world of hindgut microbes. So welcome, Dr. Belinda, and thank you so much for being here with us today.
Thank you very much, Nerida. It's a pleasure to talk to you, as always.
Yeah, we always have so much fun. This could go anywhere today because Belinda and I get together and start talking about hindgut microbes, and it's always
Never stop.
No, and there's so much we don't know and so much we'd love to know. So hopefully it'll be a really, not hopefully, I know it's going to be a really interesting conversation today. But first, Belinda, I just want to get a little bit of your backstory. Like, what initially drew you to researching the things that are invisible and literally everywhere?
Look, it goes so far back for me. So it goes right back to the end of my first year of university. I reckon I didn't even know what a microbe was.
Oh, wow.
When I was in first year uni.
Mm-hmm.
And then I went to a lunchtime lecture by a wonderful microbiologist who gave this amazing overview of how microbes affect every part of our lives. So they affect everything from the food that we grow, the health of our soil, how our guts work, how we make industrial chemicals, how we process our sewage, you name it, microbes are involved in it.
Mm-hmm.
And because of that, one lunchtime lecture at the end of first year uni, I went, okay, I know what I want to be. I want to be a microbiologist.
Oh, wow.
And until that time, I had no idea that they even existed, I'm pretty sure.
Wow, how cool. And that was back before we really knew how much of an impact they have, especially on human and animal health.
Absolutely, yeah.
Oh, wow.
So, and I guess I originally approached microbiology from the perspective of environmental microbiology. I was really fascinated by how complex groups of microorganisms got together in the environment and did things like decay leaves in the soil, and why we weren't kind of drowning in leaves after, millions of years on this planet, why there wasn't a huge pile of leaves that we were all sitting under.
Yeah.
And so that was probably
not something I've ever considered.
Yeah, well,
that's a good question.
It was a great question.
Yeah.
And it was only because of having those kind of questions put to us and then thinking about, why doesn't that happen? That was my first kind of introduction to thinking about these complex groups of microorganisms that do really cool things for us that we're not even aware of.
Yeah, yeah, wow. It's yeah. Lots of gratitude to that lecturer then in your life for introducing you to the world of microbes.
Absolutely. Yeah.
Now tell me, you as part of your business, you have, is it the zoo of poo you call it?
The Zoo of
The
Poo.
zoo of
Yes,
poo.
that's a wonderful. It's a very exciting program that actually, and I know we're going to talk about horses predominantly today, but it actually was inspired by the work that we've been doing in horses. And we sort of went, horses are really fascinating. And then we went, "Well, what else is really fascinating?" And then we thought, well, other things that are like horses, so hindgut fermenters, and we can come back to that later, but hindgut fermenters. And so we basically then started to think about all the hindgut fermenters that were there in the world. And a lot of those are zoo animals. So that sort of started our Zoo of Poo program, which we do authentic research with zoos in this area. And we involve high school and primary school students in that research as well and teach them really cutting edge techniques.
Yeah, wow. That's cool. What's the weirdest animal you've ever tested the microbes in?
The weirdest one. I don't know that any. Oh, well, maybe. Okay. So maybe the one that people would not necessarily have heard of before is the Fijian crested iguana.
Uh, yeah, wow. That's a weird
Okay,
animal.
so
Yep.
will that qualify?
Uh-huh. Yep, yep. So you know about the microbiome of the Fijian crested iguana, which would be a very niche field, I imagine.
It is. It's a critically endangered iguana, and it's only found in Fiji in the wild.
Mhm.
That's all.
Mhm.
And the only other population of it is actually in Australia, in zoos in Australia.
Wow.
So we have done quite a lot of work with one of my master's students, Blake Smith, over the last couple of years, looking at the hindgut microbiome of the Fijian crested iguana.
Wow.
But probably the one that people get most excited about at the moment is the capybara.
Yes. Oh, I love the
Everyone loves
capybara.
capybaras.
Yes. Yep. And if anyone doesn't know what they are, they're like giant guinea pigs. And quite similar in digestive physiology to a horse, interestingly enough.
Exactly.
Yeah.
That's exactly right. So that's why we thought, well, capybaras are pretty cool as well. So, but we study all sorts of things: rhinoceroses, elephants, ostriches, emus, zebras,
Oh, wow.
capybaras. I mean, all of these are actually share that physiology with the horse, in that they are what we call hindgut fermenting animals. And that really relates to the microbes are really central to energy production for all of these animals. And so all of those microbes sit inside the hindgut of these animals and help them to digest their food. And so we find that group of animals particularly fascinating because of the effect that and the importance of microbes to their digestion.
Yeah, like literally those animals don't exist without those microbes in their gut.
That's exactly right. Yeah.
So what drew you initially to researching the hindgut microbiome of the horse?
Of the horse. Okay, so we definitely are a bunch of microbiology nerds first and foremost. And when I mentioned to you before that we and this is some of my other teammates here at Quantor. So we come from that background of thinking about complex microorganism groups in the environment, but we had also been doing work on the human microbiome. And so the human microbiome, gut microbiome, is an area that's obviously very popular for research over the last few years. And so we were working in that area, but then we were starting to think about, well, what other microbiomes are sort of similar to a human? So we kind of extend our thinking in that direction, but actually much more complex, like an environment, like the forest floor, that kind of thing. And so we kind of merged those two things together. So we were really interested in looking at monogastrics, so animals with one stomach, but really interested in something more complex than humans, so that we could really sink our teeth into the cutting edge of microbiology. And so that came to us and we went, and where is it really important? What's, an animal that is really important to really understand how their gut works and how to kind of optimize the performance of their gut as well.
Yep.
And that's how we came to horses.
Yeah, wow.
So, so we've now spent, a good 6 or 7 years, I guess, actively researching the horse gut microbiome. But our interest in that has gone probably about nearly 10 years, which is the whole time that we've been running the business at the moment.
Yeah, well, it's fascinating to me that the horse microbiome is more complex than the human microbiome. And it makes sense because we have fermentation in our large intestine, but we're not reliant on it like a hindgut fermenter is. But yeah, I'd never actually thought about it so explicitly that the horse microbiome is much more complex than the human one.
It's definitely more complex. And that really comes down to the fact that horses are surviving, generally speaking, on a diet that's much harsher than a human diet. And so our foods are relatively simple to digest. And we have a set of enzymes in our body that allow us to digest key components of our human diet. And so even if we didn't have microbes, we would likely be fine with our digestion. But in comparison, hindgut fermenters, horses, zebras, and so on, really are reliant on those microbes. And those microbes, because they have such, at its heart, kind of poor quality food, we're talking like grasses and, really tough to digest kind of food. Because of that, the microbes that are helping in that digestion are much more specialized and have a much broader range of enzymes than the kind of microorganisms that exist in our gut.
Yeah, yep. So I've spoken about enzymes a little bit in the podcast prior to this, but I describe them as pairs of scissors. So and that they cut like big stuff up into really small stuff because only really small stuff can cross the gut epithelium and go from the gut into the body. So what Belinda is referring to is that horses have got the enzymes to digest proteins and oils and simple carbohydrates, but no mammal, humans included, have got the specific enzymes needed to cut up different fibres. So they literally just don't have the right pairs of scissors to do the cutting because every nutrient has a specific type of scissor, type of enzyme.
That's an awesome analogy, Nerida.
It makes it so easy to visualize when you just think, all digestion is cutting big stuff into little stuff, and you need scissors. And I often think about the hindgut microbes, and this is maybe jumping ahead of ourselves a little bit here, but I often think about us as humans or the horses sort of endogenously produce, like the enzymes that they produce for themselves as being, a couple of different pairs of scissors, and then the microbes have got like a whole knife shop. You walk into those specialised knife shops and they've got every type of shear and scissor and knife for cutting up anything you'd ever want to cut up. That's how I kind of think about the microbes, is that's what they've got access to. They've got pretty much every type of scissor that you could need to cut stuff up.
I mean, again, that's a really great analogy, and it dovetails into an analogy that I often use, which especially for fungi, which again, I know we'll probably talk a bit more about, but especially for fungi, we like to think of them as like Swiss Army knives.
Yeah. Yes.
So a single fungus has this whole
array of cutting implements.
Many, many, many different enzymes, which make those organisms in particular so powerful.
Yes.
And they really just like flip out a whole selection of knives and then chop up whatever they can see.
Yeah, well, the fungi are super cool. We'll get to them in a minute. And they're one that I don't know, or no one really knows a lot about, but Belinda and her team probably know more about them than anyone else in the world at the moment. So we'll get to them. But we keep using the terminology microbiome. Can you just give us a easy to digest explanation of what the microbiome actually is?
Okay, so the microbiome, again, coming back to what I said at the beginning, it's really the term that we use to describe the complete set of microorganisms in a particular environment. And so, where you and I are obviously particularly interested in the horse gut, the horse hindgut, and the assemblage of microorganisms that are in there. But we could say the human gut microbiome. We could say the forest floor microbiome. We could say the microbiome of the local dam. It could be the dog gut microbiome. Even when we think about it, other microbiomes as well. So there's the oral cavity of a horse that has its own assemblage of microorganisms.
Yeah.
So
and
the
stomach
microbiome.
and the small intestine, but we don't speak about them so much.
Exactly, yeah. So any location where microbes exist, it's very rare that you only have a single type of microbe. And so and when we're talking about microbes, we're talking about bacteria, we're talking about fungi, and fungi includes both molds and yeast. And then we're also talking about other things, protists, protozoa. You might have heard of some of these sort of terms before, and also viruses as well. In the end, it encompasses a huge range of microorganisms, and there is virtually nowhere in the world that only one microbe exists on its own. And so, in classical kind of microbiology, what we first learned about were when we started growing microbes in the laboratory, we would find one or two or three different microbes depending on how we grew them.
Yes.
And we would go, okay, well, that's a very important microbe. But what we've realized over the last, few hundred years and then a couple of decades especially, is that there's never one microbe in a location. It's really just about the limitations of how we grow them. And so what's happened, especially over the last couple of decades, is we have new technology now that allows us to look at microbes without culturing them.
Mm-hmm.
And so without growing them independently in the lab. And so that's technology where we can actually read the DNA sequence of all of the different microbes in an environment all at the same time.
Wow.
And that gives us a far, far more comprehensive look at what microbes are there, even things that are maybe impossible or maybe just super, super, super hard to grow in a lab. We can now kind of see them all with new technology that's available to us.
Because, of course, the ones that live in the gut, they don't like oxygen. Like
Correct.
that
Yeah.
oxygen is deadly to them. So you try to bring them out and grow them, even if you're really careful with them. It's very hard to keep things away from oxygen in our environment. So it was
Super hard.
very, very difficult to culture much out of the gut. And then I think it must have been around 2000 that this technology started appearing, and all of a sudden you could see all of these different microbes because they're looking at the DNA. And then your technology is even more advanced than most. And in fact, I said to Belinda the other day, you've ruined nearly every other scientific paper for me about microbes, because you can actually look at them down to species level. Like you can identify them by name as opposed to just looking at them on family, or I always get the hierarchy, family and genus level.
That's right. Exactly.
So you actually know exactly who's in the gut.
We do. And I mean, again, the technology is just stunning that's out there. But, even probably going back sort of 10 years ago, we would still be at the point where we're basically saying, oh, are there bacteria? Are there fungi? At that very high level. And then over the last few years, it's come to be fairly standard that we would say, is there Clostridium or is there Lactobacillus? And they are the genus level identifications of microbes.
Yep.
But then in more recent times, and certainly this is where our laboratory does as much as we possibly can, we come to the species level. And so that's saying things like well, is there Clostridium difficile or Clostridium utyricum, or so we're actually being able to identify it to an even lower level. And then the super exciting thing that our lab is really focused on is, okay, now that we know who's there, how can we have a look at their genome, at the DNA, in order to see, we're looking for particular markers of what those microorganisms are actually capable of.
Yes.
So we're looking for the signatures in their DNA of those enzymes that we were just talking about. So can we see, it's like, okay, I'll extend the analogy. We'll see how we go. Now, if you picked up the box for the Swiss Army knife, and if we read what's written on the box about what that's, because lots of Swiss Army knives are different, what's on there, we should be able to see, okay, well, this microbe has the tool for cutting up cellulose, for example. So that's what we're actually really interested in now, trying to find the signatures in the DNA for what tools those microbes actually have available to use.
Wow, that's super cool, because and this is actually a perfect segue into my next question. In what we'll go and meet the microbes in the hindgut, but within the bacteria at least, there's ones that love fibre and there's ones that love starch, and that's what you're talking about. Is if you can pick up the signature of which ones love what, then we know what the horse is set up essentially to digest in the hindgut.
That's right.
Oh, that's so cool. Wow. Okay. So let's talk through the microbes, because we have the microbiome, which you've explained beautifully as being the entire collection of all the invisible things that live in the hindgut. The group, I guess, that we've all been focused on for so long is the bacteria, because they've been probably the easiest ones to look at and the ones that we know the most about. So let's talk about the bacteria, because there's literally, like, how many different species of bacteria? There's so many, but we can put them into groups. So can you talk us through the bacteria of the horse's hindgut microbiome?
Yeah, I mean, so there are, when we look at the horse's hindgut microbiome, which we do using the manure samples. And that's a pretty good proxy for us, because the microbes in the hindgut are very close to where the poo comes out. So we can look at the poo and see what's in there and make some pretty educated guesses that's very similar to what's happening in the hindgut. And so we typically would find around about 400 to 600 different types of bacteria alone in that manure sample. And so there are a couple of kind of groups of things that we are interested in seeing. The ones we never want to see are the pathogens. So there are, a few really kind of well-known pathogens that are a problem for horses, that can cause diarrhea and so on. So I mentioned one a little while ago, Clostridium difficile. So those kind of microorganisms we really don't want to see in the horse, or we don't want to see very much of in the horse at all.
Yep.
Because I should mention that when we have a look at the gut microbiome through the manure, we can see the relative abundance of the different organisms in there. So what you want is larger numbers of the better microorganisms and smaller to hopefully no signatures of the more problematic organisms. So those kind of pathogens we really don't want to get of.
So salmonella would be one that people are probably pretty familiar with. That's one of the pathogenic bacteria, and E. coli as well would be.
E. coli is a little bit. Salmonella is much more likely to be in the harmful kind of bucket. E. coli can be helpful in some circumstances. It really depends on what type of E. coli is there.
Right. Yep.
So, and again, this is another kind of level of discrimination here in how we look at the microorganisms, but we all just say, oh, E. coli, but there's many strains of E. coli out there. And some of those have nasty toxin genes. So they actually have genes produce toxins that cause diarrhea. But a lot of them don't have those toxin genes, and some of those are actually important in our digestive tract, including in human digestive tracts, for making vitamins and that kind of thing. So E. coli is one. It really depends on the E. coli.
Yeah, right. So if you're one of the good E. coli, you're kind of cranky at your nasty relatives for giving you a bad name all the time.
Yeah,
It's like,
exactly.
it's not all of us. Sorry, E. coli. I do apologise. I didn't realise that, and I shall correct my thinking in the future. So, okay, so they're the ones that we want to see either none or virtually none of. Yep.
Yeah. hmm And then we have a whole bunch of other microorganisms in there that we don't know exactly what their roles are yet. So I have to say that there are, so as I said, there's like 400 to 600 that we can see every time we want to test. There's a whole bunch of microorganisms that we don't really know exactly what their roles are yet in there. But some of them, some of the other ones that we don't want to see too many of, but we're okay with lower levels of it, are those ones you mentioned before, Nerida, that love to eat starch in particular, or sugars, and therefore, and fructans, exactly. So we don't want too many of those microorganisms in there. And they're things like the Lactobacilli so which are implicated in acidosis in the gut. And then that through to laminitis in the hoof as well.
If there's massive numbers of them. I love, I think the first time I met you and worked with you, you called them the weedy, greedy bacteria, and it's such a good name for them because they are weedy and greedy because they do everything really quickly. And I think I was chatting with you when you were here a few months ago and said that I'd blown my own mind because I worked out that if they're reproducing, because they reproduce so quickly, don't they? These ones
do,
and some of them are every 18 minutes.
Yes.
That they can double their population.
Then things like the E. coli, they can become weedy, greedy bacteria as well, definitely.
Yeah. So if we're feeding horses a lot of, say, cracked corn that has got very poor starch digestibility further up the digestive tract, and most of that starch ends up in the hindgut, these weedy, greedy ones, the starch-fermenters, or we also will just call them the bad bacteria, as collectively as a group, their population can just explode. And if you just do straight maths and it's not, you say like they would never do this because they'd eventually run out of food or they'd overcrowd themselves. But what was the number that I worked out? It was
a crazy number.
Yeah. I'll have to try and think what it was. It was like 18 with 15 zero, quadrillion, I think, 18 quadrillion, somewhere around there, that you would actually end up with after 18 hours of them doubling every 18 minutes. So it's phenomenal how quickly they will get out of control.
It is. So luckily there are some brakes that kind of get put on those. But even where the brakes are applied, it can be kind of too late, if the organisms get too much of a head start. But really the number of organisms that are in poo, in manure, could be up to around about 10 to the 14. So that's 1 by 10 with 14 zeros after it. So it could be up around that sort of number per gram of poo. Luckily, you will never get all of those being a single type of microorganism. Again, what I've said before, even in those sort of situations, one microbe doesn't exist in isolation from the others. So the other microbes will kind of put some brakes on it. And in fact, the microbes themselves will eventually put some brakes on themselves. They'll just start to create such a hostile environment for themselves. They'll wipe each other out eventually.
Unfortunately, once they get to that point in a horse, the horse is pretty much dead because there'll be so much acid. There was some really
long
cool
before
research.
that.
Yeah, because they just get so acidic in their gut. And I think it was University of Queensland had done some research where they maybe staining. Like it's somehow the bacteria in the hindgut were coming up as different. They're not staining, but they were fluorescing.
Fluorescence.
Yeah, so they were showing like different colours as they overdosed these horses with grain to have a look and see what happens in the hindgut, and it was amazing. Like to see the weedy greedy starch-fermenters they just took over like wildfire, and then they wiped themselves out. They produced so much acid that they literally killed themselves.
They kill themselves.
Mm.
Yeah, but as you say, by the time the microbes can kill themselves, they've definitely done a lot of damage to the.
And this is the only time you'd really see that in a horse is the classic, pony broke into the feed shed and ate half a bag of feed. Like it's so hard to save them from that because there's just so much grain ends up in their hindgut and those weedy greedy creatures take over. Hey, quick moment while you're listening. If feeding your horse ever feels like a puzzle that a toddler has been playing with missing or half-chewed pieces, you are exactly who we built My Happy Horse for. The app makes everything clear, simple and tailored to your horse, so you always know when you're doing the right thing. Check it out at myhappy.horse or find it on the App Store. All right, back we go. Okay, so that's the bad bacteria, and as Belinda said, like they're a perfectly normal part of the hindgut.
Yeah,
and
as long as
they
they're
would
not
have
out of
roles.
control.
That's right. Yeah, and it's around 1% of the population is what is, I think, considered normal in a horse's microbiome. Is that about around the percentage that
For those kind of microorganisms? Yeah, I think there's a little bit of leeway there, depending also. I mean, certainly you definitely don't want too many of them, but it also depends on what other microorganisms from the good guys are in the gut as well. So they kind of balance each other out.
Mm-hmm.
So the good guys, I guess, turning our thoughts to those, the ones that we're particularly interested in horses are the fibre-degrading microorganisms. And so there are a couple of fibre-degrading bacteria that we're particularly interested in. So there's one called fibrobacter, and there's another one called ruminococcus. And just to sort of clarify, I mean, horses are not ruminants, but the reason why ruminococcus is named that way is because it was a bacteria that was originally found in cows, ruminants. But it, just like horses, cows eat a lot of grass. And so cows, although they have a very different digestive system, they have some overlap, with horses in the type of microbes that are there because they are eating a common kind of foodstuff. So things like ruminococcus and fibrobacter are microorganisms that are bacteria in particular that are able to help with degradation of those hard to eat kind of fibers.
Yeah. So for horses, a horse's diet is predominantly forage, which all horses' diets should be based on and dominated by forage, so pasture or hay or different haylage. There's lots of different forages you can feed, but those good bacteria are the ones that have got the scissors to cut up the fibre and give. And they sort of, my understanding is that very broad understanding is that they cut the fibre up and eat it and they ferment it.
Yes.
And then they essentially give the horse their leftovers. These are the bits we don't want.
That's right. Exactly.
And those leftovers are in the form of volatile fatty acids, which the horse then absorbs and uses. It's a beautifully designed system.
It is a beautifully designed system. So, and then beyond those bacteria that can degrade fibre, the really kind of exciting new cutting edge sort of stuff is looking at what other organisms other than bacteria are involved in that fibre degradation. Because although those bacteria have some of those, they have a small Swiss Army knife.
Mm-hmm. They've got a pair of scissors and a knife.
Correct.
The very basic Swiss Army knife.
Very basic, better than most.
Yep.
But in comparison, what we're really interested in looking at and what makes the horse gut so different from the human gut is really the presence of fungi as well.
So we don't have fungi.
So we have very, very few. So really the one that gets a little bit of talking about is something like candida, so which is a yeast. So it's like one of the most simple forms of fungi. And that is something that we actually don't want really in our guts normally, but it is a normal part of human guts. But like they're also like a weedy greedy sort of thing
Yeah.
in our guts.
Okay,
And
in
we
small
don't want
numbers.
too many of those. But horses are far more complex than that. And they have much more complex fungi, which are a type of mold. So yeast, single cells, molds, multiple cells. And so molds, which we just they are a type of fungi. So molds are something that the equine hindgut has, and all other, so zebras and so on. Those are also environments where we find those kind of things as well. And then the particular type of fungi that we're interested in are called anaerobic fungi, because as you said before, Nerida, the hindgut of horses is an anaerobic environment, so not very much oxygen at all, normal circumstances. And that's where these kind of microbes are able to flourish. And anaerobic fungi have like the premium U-Butte pocket knife,
Yeah.
Swiss Army knife there,
Yeah,
with all of the tools, like a great big arsenal of tools to apply to those things like cellulose, but also hemicellulose and lignin as well, which are the tougher parts of plants that when the horse is grazing, foraging, it's taking those up all the time. And so there are actually these specialized organisms in the hindgut that can help with breaking down even those really tough molecules and freeing up, breaking them into smaller molecules that then free up and eventually are able to be digested by other bacteria as well in the hindgut. So they all kind of work together as a community to ultimately deliver that energy, the volatile fatty acids and so on, to the horse.
So the fungi, my again basic understanding of it is they have like tentacles and will grow like out and through the fibre and do their cutting. So they're sort of taking the big stuff, really big stuff, and cutting it down into smaller, simpler stuff, and then essentially feeding the bacteria that smaller, simpler stuff, and the bacteria will take it from the fungi.
That's right, exactly. And what you just sort of was talking about there, so tentacles is a good way of visualizing it. We call those mycelia. And so they, if a fungus is on, I mean, just think of your bread going moldy. Okay, so how that all starts is one little tiny spore falls on your bread. The spore the spore of a fungus goes, oh, this is a good place, so I might explore this. And so then you sort of get the little spore growing outwards, and that's mycelium spreading across your bread. And here's a little thing. When your bread starts going green, the mycelium is already spreading, but the green is actually, it's had so much to eat, it's now forming new spores. And it's actually the spores that are the green bits.
Oh, there you go.
The mycelium is typically white. But that happens.
So I was told years ago, and this has got nothing to do with horse nutrition, but I was told years ago that when you can see the mold on bread, that the mold is already completely through the bread. Is that?
Yeah, it certainly will have spread a long way by then.
You can't just pick the moldy bits. Not that I do this, but you can't just pick the moldy bits out, and it's grown for so long in that bread that it's actually forming spores by the time you can see the green stuff.
That's right, exactly. And so we typically would not advocate for people to just cut the mold away and eat the rest of it. And that's because most of the time you don't know what that mold is. And unfortunately, in the world of fungi, these things can produce, I mean, everyone would know about the idea of mycotoxins. So that's toxins produced by fungi. And we're not talking about anaerobic fungi producing mycotoxins here. These are different types of fungi, but those kind of ones that, fall on bread and cheese and, I don't know, lying on the top of your sauerkraut and that kind of thing. You don't really know if they're a mycotoxin producing one or not. They're probably not, but for the sake of a bit of cheese, would you really kind of risk it? So
Yeah.
get
rid of the moldy bread and the cheese.
Get rid of the mold.
So yeah, the fungi in the gut obviously don't. Well, the good fungi. I guess we don't know enough about the fungi to know whether the gut can get populated with toxin producing ones. Do we? Is that a possibility or not really?
Oh, look, I mean, it's unlikely because as far as we understand at the moment, anaerobic fungi are not producing mycotoxins. Type of fungi that do produce mycotoxins are ones that are more likely to grow in the presence of oxygen.
Yeah, right. That makes sense.
So for horses, it's much more likely that, because we do know mycotoxins are a problem for horses, but it's about pre-formed mycotoxins in the forage or in the food, the grain, and so on. So rather than those fungi growing in the gut, because the conditions in the gut shouldn't be conducive to those fungi growing. They're conducive to anaerobic fungi growing, but not those mycotoxin producing fungi growing.
Yeah. Okay. That's interesting.
So that's a good thing. A good thing.
Yeah. So
and nature at its best,
yes.
not making sure that there's a bit of a delineation there between those two groups. So,
yeah. One survives with oxygen and the other one one survives with oxygen and one survives without oxygen, but they don't cross over.
That's right. Yeah.
Fascinating. It's such a fascinating area. So we've got in the gut now. We've met the pathogens, so the disease-causing bacteria that we don't want in there. We've met the weedy, greedy, starch-digesting bacteria.
Sugar, fructans,
that will explode and get out of control when we feed our horses the wrong thing. We've got the good bacteria, so the bacteria who will digest fibre, and then their friends, the anaerobic fungi, who help them to go through that process of fibre digestion. And then the fascinating bit then is in a lot of those unknown ones, because we know that there seems to be a gut access to every part of the body now. Every time I discover a new one, I'm like, it's not surprising, but I recently discovered the gut-bone axis. So they're discovering, that there's some sort of interaction between gut bacteria and osteoporosis in humans. I'm like, oh, great, add that to the list. But it's an interesting one for me as a horse nutritionist, looking at bone health in young horses and whether gut health has an impact on that. But a lot of those unknown ones potentially would have roles in immune function and behaviour, like the gut-brain axis, which is probably the axis we understand the most about, even though we still understand so little. But what else might those unknown ones be doing? Have you got any thoughts on that?
Yeah, I mean, I think you've picked off a couple of the key ones there. Definitely immune system function. So metabolism, metabolic function generally. So just glucose supply to like skeletal muscles and so on. So definitely very important. So you mentioned the gut-brain axis, and that is just starting to really be studied in horses. So and how that might interact with anxiety and stress and behaviour in horses. It's certainly something that's been still only just touched on in humans. And I mean, the reality of things are we tend to investigate ourselves more to begin with before we start to investigate anything else. That's why we threw the Fijian crested iguana in there, Nerida, because we kind of went, well, who's ever going to get to the poor old Fijian crested iguana at the end of the road?
Yeah, I don't imagine there's a lot of research dollars being put towards that little creature.
Brought him forward a little bit, but we know a reasonable amount in humans now about those kind of things: immune function, gut-brain axis, metabolic function. But we're really just scratching the surface in horses at the moment.
Yeah. But they produce, like, a lot of our human dopamine is produced by the microbes in our gut, isn't it? And there's all sorts of other neurotransmitters that they produce. So we're really cactus when we mess up our gut health. We really
Yeah,
are. Mentally and physically, we're just
we are.
ruined.
And one of the most amazing things I know, and this was about, oh gosh, about 5 or 6 years ago, I guess I learned, was that there's and I honestly can't remember the exact interaction at the moment, but there is a microbe in the human gut that when it wants to get when it wants to compete against a different microbe in the human gut,
Hmm.
it actually sends a signal, if you like, to the human and gets the human to do the dirty work to get rid of the other bacteria by changing something in our physiology.
Ah,
And so so microbes both cooperate and compete with each other all the time. And that balance can really shift. Even with the same microbes under different circumstances, that balance can shift. But then you put something like the human in the middle of that, or the horse in the middle of that, and the number of kind of interactions and the complexity of those interactions. One of the reasons I love microbiology so much is I could be alive for the next 300 years and still not answer 1 millionth of the people are out there, which is kind of frustrating in a way, but also kind of really cool because you
exciting.
can just keep going forever.
Yeah. I heard someone say the other day, I don't have all the answers. I don't even know what all the questions are yet. And I was like, well, that
100%.
that applies massively here. So
Yeah.
I think you've sort of just touched on it, and I wanted to just bring it up briefly today. Can you just explain to us, because it literally blows my mind and fascinates me, the quorum sensing.
Yeah. Yeah. Quorum sensing is very cool. In a previous life, I thought, oh, I'm going to be a quorum sensing researcher, but that hasn't been my calling so far. But it is a totally fascinating thing. And so the high level view of that is, microbes they're not sentient like us. They're not. We anthropomorphize them more. Excitable microbiologists like me do. We talk about them thinking and doing things and so on, but they're not really like that. They're existing, but they have this freaky thing called quorum sensing, where they are sending out little chemical signals into the environment. And when those chemical signals build up enough, it tells the bacteria, because that's mainly where quorum sensing has been studied at the moment, so I'll say bacteria. It tells the bacteria that there is a certain number of those same type of bacteria all together in the same location.
Mm.
So it then is able, once they sense that there's a certain number of them in the same location, they then tend to change the regulation of their genes.
Huh.
And so that means that they then might turn on certain enzymes that they
Wow.
wouldn't bother to do if there was only 1 or 2 of them around.
You're right.
So the way I kind of sometimes think of it is, it's like if you go to, a big concert in the stadium. And, if there were 2 people in the stadium listening to the concert and they held up their mobile phone with their light on,
Yep.
it would be pretty naff,
Yeah.
right?
No one
Um,
would notice.
but if you have a whole stadium full and they're all turning on their light on their mobile phone, and then there's this natural inclination that everyone in this is going to start doing this. They're going to start waving back and forth, right? Because it's just, they've got the signal. The signal has gone up. That's the mobile phone being lighted. And then there's just this natural inclination in humans, right, for whatever reason, that we're all going to start swaying back and forth.
Yep.
It's like that.
Okay.
So they send out the little signals, and when they can all see the little signals, they start doing something in concert together.
So give us an example of something that might switch on. Have you got an example?
They might switch on a gene to make a protein, an enzyme that degrades something, some sort of substance that they wouldn't bother with normally. And so that may be, for example, just like kind of humans, right? So if we've got a bowl of lollies in front of us or a salad in the fridge that we could go and get, and I'm saying that because I have a big bowl of lollies in front of me, but I'm being very restrained. We would tend to grab a couple of lollies before we'd bother to go to the fridge and make a salad, right?
Yes.
And so bacteria are the same. They might be in an environment where there's a little bit of glucose floating around. And so they might say, oh, well, I'll just snack on the glucose and I won't bother making these particular enzymes
Interesting.
to attack something that's more complex than that. Because there's a downside to using your energy, make the enzyme.
Yeah, because enzymes are a thing, like they're made out of protein, so they actually take energy and protein to create them. So it makes sense if they don't need it, then they wouldn't make it.
Exactly. And it may be that they need quite a few of them to contribute to that before it's really worthwhile to do it. So that may be that kind of thing that happens. The other one that's pretty classic is that they may know when there's enough of them present, if there's another microbe that kind of competes with them in that space, enough of them present, it's a little bit like mounting a bit of a military campaign here. If there's enough of them present, they might start to say, all right, great, we've got the advantage now of numbers. So let's turn on, for example, some sort of toxin that's toxic against the other microbe.
Oh, wow, and take them out.
And wipe that one out.
Wow. Yeah,
so they do all this kind of complex signaling, but it blows my mind still as well. There's that obviously run in the bacteria. It's like AI or something. You just kind of, the button gets pushed and they go off and do it, but they're not really thinking about it.
They're driven by survival like every other living organism, I guess. So it's all survival mechanisms that they've developed over how many billions?
Millions and millions and millions of years. That's it. They've got a little script that they run and off they go.
Yeah, because it'd be close to how long have bacteria been on the earth?
Nerida, you would ask me that. I don't know.
I need to ask my 12-year-old son that. He would be able to give us that. He's my many millions and billions of years ago expert. So
that'll be my Google thing.
Yeah.
How long? Hang on, I'll Google it for you.
So where my mind's just gone when you were talking about if they had a ready supply of glucose, so the analogy of if we had a jar of lollies versus a salad in the fridge, we'd go for the easy thing, which, human nature, we would all do that. Even like as nutritionists, I'd like to admit that I wouldn't do that, but I'm human. But in the horse's gut, what I see in horses that are fed lots of grain, so a high grain diet that is going to be putting starch into the hindgut and then favouring those bacteria that are the starch-fermenters, the weedy, greedy ones. If they're sensing, I guess, that, there's a lot of them and then they've got a ready supply of food, I mean, they can live on fibre, but if they don't have to do the hard work of cutting up fibre, then they're just not going to.
Yeah.
And what we see with these horses is that they're what we call a hard keeper. Like they take so much feed to keep weight on. But part of that would be, and I've always thought about it as that the weedy greedy bacteria just outcompete and kill off the good bacteria, which is a big part of it, I think. But maybe also there's that going on where they just go, well, there's so much of this starch stuff that we're not even going to bother producing the enzymes to digest the fibre.
Yeah.
And reducing fibre digestion in that way as well, which makes them really hard keepers.
That is definitely the case. So most bacteria, most fungi as well, can use a variety of, we would say, substrates to grow on. But typically speaking, so they'll have kind of overlapping groups of substrates that they can use. But they will typically have one or two that are their preference. And if those are available, they'll get in and chomp those up. Now, some bacteria, some fungi have evolved in a way knowing that there's a whole bunch of things over here that will eat those things if they're available. And so they actually evolve not to eat those things. They evolve to eat more complex things where there's not much competition. So, but definitely under some circumstances, you can have both rapid growth, allowing for that weedy greedy kind of thing going on, but you can also just have, I'll just eat that because it's easy.
Yeah.
And I won't bother to do anything harder than that until I run out of the that
Yes.
easy stuff. Till I
Yeah.
run out of the bowl of lollies, and then I go, oh, well, I better go make the salad
Yeah.
then.
That's right. When you get really hungry and desperate for something else to eat. So you've just led us into another really fascinating area in hindgut, the hindgut microbiome, in that we're now starting to understand how important diversity is. So,
Yeah.
you've mentioned a few times that there will never just be one microorganism living in there in isolation, but what we're starting to understand in multiple animal species is that the more microorganisms we have in the hindgut for the horse, the better. And you just led us into why that is, because you mentioned that each microbe will have a favourite food.
Yes, that's correct. And yeah, look, we know from human studies, we know from horse studies and also of other animal studies as well, that diversity is important because of that. Again, we'll go back to that analogy. So now we've got multiple Swiss Army knives all happening at the same time. And so that's good. It's making the most use of the resources available. But that is an important thing that up until fairly recently, microbiologists have been able to only really measure diversity on the basis of which types of microbes are there. So that's saying, well, have I got Clostridium difficile, and have I got Lactobacillus, and have I got Fibrobacter, and have I got Ruminococcus? Because, for example, that tells us, okay, we have a diverse group of microorganisms there because we know them by name.
Yep.
But now what we're moving into the era of is, rather than caring what their names are so much, it is about, well, can I actually look at what's on all of the boxes of all of the Swiss Army knives, and what is the diversity of enzymes that can be produced by these. So, because that is really where the heart of it is. It doesn't so much matter, if I've got a broken sink and I've got a whole, I've got 20 people in front of me. It doesn't really matter what their names are. It matters if there's a plumber.
Yeah. Yeah. Yeah, true. Yeah, that's a really good analogy. Yeah, you just need someone to fix your sink. So it's the enzymes for digestion, which is I guess back when I did my PhD, which now when I look at it, looking back 25 years ago, it was about how do we get starch digested in the small intestine so we don't change the hindgut microbes.
Yeah.
But we looked very much at them. We knew that if we fed too much grain and shifted the microbes, that we would change a horse's behaviour. That had been published, but there was no gut-brain axis. Well, there probably was, but it wasn't. I hadn't come across it at that time. But the diverse microbes, they'll produce lots of different types of Swiss Army knives, so they've got all the different enzymes they need to digest fibre. But then you'd also be able to have a look at what hormones and neurotransmitters and things that they would produce, like all that other stuff that they do. That's as important. I mean, if the horse can't digest its fibre, then it's in a really bad place. But if it doesn't have the right microorganisms to, support its immune system or control its immune system even, and produce the neurotransmitters and hormones to affect behaviour, then it's in a world of hurt as well.
Yeah, that's right. And that again is why we need to be around for the next 300 years to invest these things.
I'll sign
because
up for that with you.
Keeps going on and on and on.
Yeah.
And it gets more and more complex, unfortunately, as well. The more that we do that, we want to go into that area because, you're then looking at really combining a lot of different types of scientists in a room to solve those kind of problems as well, because you're looking at horse physiology and horse nutrition and microbiology and immunology and it just keeps going and going.
Bigger and bigger.
It's enormous, that bit.
The cool thing, and I think there was a couple of papers published last year that made people in the horse world sort of really sit up and listen, particularly amongst my clients, or some of my clients who are very racing industry focused. And that was a paper by Park and another by Lang and their co-authors. And they both showed that the more diverse the microbiome was for horses, the better those horses' performance. and Park, I think, showed that in thoroughbred racing horses in Jeju, I don't even know if I'm pronouncing that correctly, but a different breed of horse that I'm not familiar with, but the horses with the higher microbiome diversity were also the horses that performed better in racing. The Leng paper was freaky because they showed that at 28 days, the microbiome diversity at 28 days was affecting performance at 3 years of age.
Yeah, wow.
Like, it just is mind-boggling. And so it's been a really good thing because people are now thinking more around, well, it's affecting our horses' health and welfare and ability to digest food, but it's also affecting performance.
Yes,
and
that's
then
right.
that in my world is, well, how does that, how do we bring that back then to how do we feed our horses to make sure we get that microbiome diversity?
Yeah.
But I think probably the most fascinating thing for me that's come out of all of that and the really, does it really have to be this way? Who designed this system and can we change it? Is that it's actually the mother,
Right.
like the dam. And I'll get you to explain what you know on this because I only know just enough to be dangerous on this one. But the dam that essentially gives her foal a blueprint of which microorganisms are good and which are not. Can you explain that really inconvenient piece of science to us, please?
And it again, it's a reasonably common thing with hindgut fermenting animals as well that happens. So first of all, the key thing is, where does the horse microbiome kind of come from in the first place? And a lot of that is to do with during birth and during the first few kind of days, weeks, hours of the horse or the animal coming into being, that they are picking up their microbiome from the local environment. And that is basically their mother that they're picking their microbiome up from. And so it's really important then that a foal would be exposed to a good microbiome to start with. And you were mentioning before about sort of hormonal signals and so on as well. These kind of signals may actually influence and be passed on through the mother to the foal in order to pre-select the kind of microbiome that is beneficial for the foal in that particular environment, which again is linked around the mother as well. And so
that's happening in utero though, like before the foal is born. Yeah.
Exactly. So we know that there's lots of, again, chemical signals, hormonal signals going around while the foal's in utero. And so we think that this is super new kind of research.
Yeah.
We think that may be predisposing and creating the kind of, let's say, chemical kind of accepting environment for certain microbes to be pre-selected and to be remain associated with the foal in those first sort of 28 days or so of its life.
It makes a lot of sense of different things that we see in the field, and also research that's been published previously. Like there's a paper in ruminants in dairy cattle where they take the rumen contents from a number of cows and swap them around. So they wash the rumen out as much as they can in the cannulated animals, and then they'll take cow A's rumen contents and give them to cow B and swap everyone's. And within a matter of weeks, those cows had all shifted back to virtually what their original microbiome picture was. So I think of it as the dam saying to her in utero foal, this one's okay, this one's okay, this one's okay, that one's not. That one's not, that one's not, this one's okay. But it must be based on what her microbiome is at the time.
Correct.
So if hers is unhealthy, she will then train her foal to have an unhealthy microbiome, and that's not something that we can change later in life, which makes it really inconvenient.
It's quite hard. And I mean, again, that thinking is also coming out of human studies as well. And which has also been informed by some mouse studies and so on. But So the establishment of that microbiome can be quite hard to shift. It's not impossible to shift, and we would all, as humans especially, and horses definitely, we would all benefit from having better conditions in our gut, not eating so many lollies, eating more salad and so on. And so I do want to say that can shift the balance of microbes in your gut. But having said that, there's this kind of concept of enterotypes, so that you might be predisposed to having a certain predisposition to having a certain group of microorganisms or that.
So your pattern
And
will
that,
look like this as opposed to
yeah,
that person whose pattern looks like that.
exactly. And it can shift more towards one pattern or another, but you're overcoming then multiple kind of hurdles that have been sort of set up over time, which is to do with just how those microorganisms that you started life with are interacting with other microorganisms, maybe going back to our quorum sensing and so on, keeping other microorganisms at bay. If they've got a foothold there, they don't want to let anybody else in to compete. Um, and and that taking up some early real estate in the gut is an important thing. If you claimed your real estate and you're kind of good at defending it, I feel like I'm venturing into the home market now, but if you've got your real estate,
You're going to
you're
want to
kind of
defend it.
set,
Yeah.
and you can kind of keep everybody out of the market.
Yep.
So that there's a lot going on in there, but that's what we think is happening to give the tendency, even with that study you mentioned about washing the microbes out and so on, there'll still be microbes in there, hidden in little crevices and so on. And so, and if they're just naturally adapted to that environment over a period of time, and they know all the tricks and tips to get rid of everybody else and to maximize their use of the environment. And that again comes from chemical signals from other bacteria, chemical signals from the horse.
That's right. The host must be just have a big influence on who it
Huge
allows in there.
influence.
Because we see there's a condition in horses called free fecal water. So it's not diarrhea. They have normal fecal balls, but then after all of that has come out, then you get like just this runny water come out after it. And it doesn't seem to affect the health of the horse. It's more an inconvenience, and it doesn't do their skin down the back of their legs much good, and their tail's always covered in manure and stuff. So it's not a nice condition to have. And they find that doing a microbiome transplant or a fecal transplant, so through nasogastric tubing in horses to put a healthy microbiome back in, has a positive effect for a short period of time, and then the horse will revert back to free fecal water. So it's like you can put a healthy microbiome in and it'll exist for a little while, but then the horse reverts to what it was previously.
Exactly, yeah. And that's some of what we know with probiotics as well. And again, coming from humans, there are very few probiotics, I can say I'm not aware of any, that you can just take once and it's all good.
Yes, because they don't establish themselves in the gut.
No.
Yeah, right.
But if you're taking probiotics on a kind of continual basis or fermented foods on a continual basis, apart from the fact that they also may be immunostimulant, or they are immunostimulatory as well, which is a good thing. But those microbes are just not set up to compete with the things that already live there. They can kind of go and knock and maybe stay in an Airbnb in the gut for a while and that kind of thing. But eventually if you stop taking them, they're moving out. And the normal owners are going to still occupy that space.
Yeah, that's a good way to think about it. So where are we at? So people sometimes want to see what microbes are in their horse's hindgut. Where are we at from your perspective with microbiome testing?
Yeah,
in the horse industry.
So we can definitely look at what's in the microbiome. At the moment, though, mostly what we can look at is bacteria in the microbiome. And that's kind of for a couple of reasons. Bacteria are actually generally much more studied in the world, like in every environment. Bacteria are like the humans, of the microbial world. Okay.
Yeah.
So we study humans a lot, and then we study all the other animals. We study bacteria a lot, and then we study all the other microbes. So we can, through this DNA kind of sequencing and so on, we absolutely can now have a really good look at things like anaerobic fungi and protozoa and so on. But for some of those microbes, we don't yet know enough to be able to tell you perfectly what that means. Okay, if you've got lots of these organisms or lots of those organisms, we can't necessarily tell you exactly what that means. Because, as I said before, we don't know exactly what every microbe does in the gut at the moment.
Mm-hmm.
But we can definitely test for them. And certainly in seeing the types of microbes that are there, we can see the balance of microbes that, especially when it comes to the bacteria, we can see the good and the bad kind of balance. And we're getting closer and closer all the time to seeing that unfolding of all the Swiss Army knives and being able to tell what these microbes are going to be able to do in the gut. So I guess a key thing would be looking at if we're testing the gut microbiome, do you have a good percentage of fiber degrading organisms? Is your diversity really good in your gut?
Yep.
Do you have too many of these weedy and greedy kind of microorganisms? And they are the sort of things that we're routinely testing for now.
So when would you suggest someone checks the horse's microbiome? Have you got? There's probably more a question for me. I need to ask myself that question. But have you got any, like, from the work that you have done? Have you got any situations where you have found that it's really useful for someone to test?
Yeah, look, I think in terms of useful for testing, definitely if you're really interested in, first of all, let's go to performance horses. Okay. So if you're really interested in improving the performance of your horse, be it in whatever kind of discipline, really understanding how the gut microbiome is working and how any interventions that you're doing are having an impact on the microbiome is definitely worthwhile to have a look at.
Mm-hmm.
And that, especially obviously in nutrition, is important. It may also extend to things like stress, though. Transporting horses a long way. We have done some studies which show that for endurance horses, if you're transporting the horse and then they're doing the endurance event and then they come out of the endurance event, transported back home, we've shown that even after 3 weeks, the gut microbiome is not back in balance.
Yeah, well.
And so, thinking about things like, well, how do you actually time your horse's events so that your horse is at peak kind of metabolic capacity?
Yeah.
That's the sort of thing that is, really reaching and forward looking. We kind of call front running in microbiology and microbiome research.
Because presumably if you competed a horse and knocked its microbiome around and then competed again before that microbiome's had time to recover and then do it again, you just like on a downward spiral, really.
Exactly. Exactly.
Yeah.
So I think that there's real value for performance horses in looking at that.
Understanding.
From a more general side of things, obviously, if you're, and of course, we're not vets, so of course you would always be talking with your vet or your nutritionist, equine nutritionist, about the best kind of regimens for your horses. But if you are really interested in the nutrition of your horse and seeing the effects of those different dietary changes on that horse microbiome, that's another good reason to test, especially if you're doing all the right things and the horse doesn't seem to be responding, because
we've seen this in some of in little bits of the research that we've done, well, I've been involved on the periphery of, but, you see horses that they can all be on the same diet, and yet they have vastly different microbiome structures. And we were picking out the horses that had poor performance based on their microbiome. It was pretty fascinating.
Exactly. Yeah, that's right. So, so seeing, if you're doing the right things and it's still not seeming to be working, it's useful to have a look under the bonnet and see what's really going on in there. And you may have, we were talking about that resilience of the microbiome, even though you're doing all the right things. Maybe you've got a really recalcitrant kind of microbiome there. You're having trouble shifting. And so again, having a look at that and seeing if there's something that stands out. Now, what you can then do about that, we're still trying to figure that kind of thing out. If you've got a super recalcitrant biome, you're doing all the right things, they're just going to be problematic forever. If you're in the performance stakes, this is where thinking about the microbiome kind of earlier in life is probably really important.
Yeah, absolutely. And I think for me, that's probably where microbiome testing would be most useful, because so much breeding happens these days with recipient mares. So we're taking standardbred mares and thoroughbred mares out of the racing industry, and then using them as recipient mares for Australian stock horses and quarter horses and all sorts, like every breed really that can breed with artificial means. So if you happen to pick up a mare that has a microbiome that's not healthy, then that's what you're breeding for your very expensive, very genetically well-bred foal. She's not, that foal's not going to take on the microbiome of her natural dam, we would presume. She's going to take on the microbiome of the recipient.
Exactly.
So for me, I would never breed a foal out of a recipient mare unless I'd tested that mare and knew what that microbiome was.
Yeah, I think that's a really very good use case for the kind of technology now that we have available to us. And of course, on top of all of that, certainly, and this would obviously be in consultation with vets, if the horse is suffering diarrhea constantly and so on, definitely having a look at the microbiome, the balance in there, and looking for pathogens in there as well. Obviously, vets are set up to do that testing in a certain way, but there are these new technologies now available that can really shed light on that. And I understand completely. I don't have a horse myself, but I know a lot of people that do, and I understand completely that very important relationship you have with your horse. They're with you for so many years. You must do the best that you can for them. And so, this is a tool that's now out there, and you might not know that it's out there, that your vets can kind of call into being as well.
It's honestly the first time I saw the data that Belinda's lab produces, I said to you, this is the closest thing to a horse talking to me that I've ever had. Because all of a sudden I could see exactly who was in its hindgut, and you could make sense then of, like, there was some cases where we were told, oh my goodness, the behaviour of that horse, so unpredictable, so difficult, and looking at the microbiome, like we don't really understand why the particular bacteria and fungi in that horse's microbiome were having that effect on behaviour, but you could see that particular horse, or there was a couple of them, had a vastly different microbiome to what we would consider a normal looking microbiome. So it's it really is amazing. And I think it would help to make sense too if there was a weanling on my last trip to Japan, and amongst hundreds of other weanlings, and this particular one had diarrhea its entire life.
Yeah.
And they were asking me why, and I'm looking going, well, it's on the same feed. Like, it can't be the hay, it can't be the feed, because if it was, then there'd be at least others, not necessarily all of them, but at least others that would be affected. And the cool thing about the Japanese farms that I work with is they have records on everything. They can tell me at any point in time what, that foal's mother was fed and any treatments that she had and all of that thing. But when I was asking them, was the foal treated with antibiotics when it was a foal? No, they looked, it wasn't. Did the dam have antibiotics when she was pregnant? And initially they said no, and then they went, oh, hang on, this mare had colic surgery when she was,
Oh,
it was
right.
around 3 months pregnant, so she would have had so much antibiotic,
Absolutely.
which
Yeah.
you need to under those circumstances. And I can't help but think that all of that antibiotic treatment during pregnancy has changed that programming that the foal has received, and he's been received to have a program to receive a microbiome that is not necessarily very healthy, and then he's had diarrhea all his life.
And that again is a very good point, Nerida, about, the impact that antibiotics or any sort of antimicrobials can have on the gut. Of course, you need to have those things when the horse's, life is in danger and so on. But understanding then how well your horses bounce back from that in terms of their gut health is another, and definitely another area where this kind of technology can be applied. Certainly, again, in the human world, I keep referring to this because there is just far more studies in humans, but there are well-documented cases of humans having antibiotics for major things and just never really bouncing back, without some really targeted kind of interventions to get that microbiome back and functioning. So so that's another. And I guess then ultimately as well, what will happen is the more that we investigate this, the more data that we accumulate about what does a healthy hindgut look like for horses. As scientists, we pull together data from around the world, the different papers that you mentioned, the different journal articles that you mentioned, we pull that sort of data together. We have our own research as well that we add to that. And over time, that accumulated data really starts to map out what a healthy hindgut looks like. And so, if nothing else in the world, adding something to the canon of knowledge that we have about our equine friends is always a useful thing as well. So people can sometimes do things just for the joy of getting some knowledge as well. So and I know you and I do that.
Oh man, it's so fascinating. I'm looking at the time thinking, Farah, we've been talking for so long and we really need to wrap this up, but there's so much more we could talk about. But we have just thrown a lot of information in a reasonably short space of time. But for anyone who is feeling like that is a lot to take in, what is the simplest piece of advice you can give that would help horse owners to maintain the microbe health in their horses from the microbes perspective?
From the microbes perspective, it would be give them a good diet. So give a diversity of feed, forage, and because we know that there's a link between the diversity of food that you present as a buffet and the participants that are going to rock up for that in terms of the microbes. So diversity in will establish diversity in the microbiome, and that opens out all those Swiss Army knives. It means that you get more efficient use of resources, and so more energy production. And we believe, a more diverse group of chemical signals and so on that can add to metabolism and immunostimulation and so on. So I think that's the most important kind of thing that I would say out of our research so far.
And it's something that we haven't mentioned specifically, I think, too, is that the more diverse the microbiome, the harder it is to knock out a balance.
Yes,
which I think is really
right.
important.
Yeah, exactly. Yeah. I can't think of a good analogy for that at the moment. I'm racking my brains. I like analogies a lot, but yeah, that is definitely and it also means that diversity also means that you'll usually have some redundancy in the group of microorganisms that turn up for the buffet. So you'll probably get more than one guest able to eat different parts.
Yes.
And so that adds to the resilience of the microbiome as well, because even if, you've got two microbes with the same tool, but they'll have, different other things that affect them. So, but if you knock one of them out accidentally for some reason, there's another one ready to go. So that's what diversity can also bring you, redundancy in that microbiome as well. So yeah, all
So
of
in
that.
the situation with the endurance horse that you mentioned earlier, like an endurance horse that's fed, say, one type of hay and then a hard feed will have
Yeah.
a less diverse microbiome than an endurance horse that is out grazing on, very varied pasture and then is also getting some legume hay and perhaps another couple of types of grassy hays and maybe some alternate fibres. If those two compete, you would expect the one on the very diverse diet to have much less disturbance in the hindgut than the one that has the less diverse diet would have more disturbance and therefore longer recovery
Exactly.
times.
Yeah,
Mm.
that's
Oh,
right.
it's also fascinating. Honestly, I can talk to you for hours, but we do need to finish up. But are you willing to participate in our clear round with no refusals, Belinda? Clear round of questions with no refusals. Well, you do, but so I've got a question here. I don't know if you have one of these, and you can actually pass. You can refuse on this one.
Okay.
What is your all-time favorite horse movie or book?
So I debated on this. I think they're the classics, but certainly Black Beauty is the book.
Oh, yes. Yeah.
But the film is actually National Velvet. It's an
Oh,
old movie
yeah.
now, from the 1940s or whatever. But I think that it's, got Elizabeth Taylor in it when she was very young, and my mother loved Elizabeth Taylor, and I think my mother really wanted me to be Elizabeth Taylor and maybe ride some horses. So I ended up going to a lot of horse camps when I was young.
Did you?
Yeah.
There
So,
you
and that's all
go.
because of National Velvet. So
Oh,
I think that's the one for me. I mean, I as I say, I don't have a horse, but I do love them. They're magnificent animals,
they
and
really are.
yeah. So that was my exposure growing up. Off to horse camp to try and be like Elizabeth Taylor in
Oh,
National
that's
Velvet.
such a cool story, and I must watch National Velvet with my daughter. I think she would
It is a
love that.
lovely
So
movie.
thank you for
Yeah.
reminding me of that one. Yeah, it's been a long, long time since I've seen it.
Yeah.
What is one myth about microbes you wish would disappear forever?
Oh, look, I think the one that I would have usually said is that all microbes are bad, right? But I think we're going well beyond that these
Yes.
days. I think we appreciate that microbes are good. So it's not so much a myth, just an annoyance. I really wish that when people on the media talk about microbes, they get it right when they talk about viruses and they talk about bacteria, because they're always mixing them up. They're always talking about, this bacteria norovirus. And I'm like,
uh,
it's in the name.
yeah.
It's a virus.
There's a little clue there. Yeah, right. Yeah, that would be. It's funny, isn't it? What frustrates us? Like, I think I've noticed things like that, but I don't dwell on it because
I know.
it's not my field of expertise. But when it's your field of expertise, that would be incredibly frustrating.
And when it's in the name,
Yes. Yeah. It's not like it
I
was
can forgive
difficult.
it if it's not in the name, but yeah.
Okay, if the critters in the hindgut formed a rock band, what would they call themselves? Who would be their lead singer and why?
So I'm going to go hard rock here,
Yep.
bordering on heavy metal.
Mm-hmm.
I think it should be Pyromyces. Would
A
be
fungi.
the name of the band, I think.
Oh.
So because, or maybe the singer, I'm not sure. But Pyromyces, because it sounds like pyro, like fire.
Oh, yeah.
I can just see that stage set up with all the little microbe rock stars on there. And Pyromyces, which is one of the anaerobic fungi, just lighting up the whole stage with those kind of, massive heavy metal kind of fireworks or something.
And they are becoming the stars of the show, aren't they? The fungi.
They are.
Like
And then the other one I've
to
got
learn
is,
there.
but this is not so good because of that naming issue, but the Rumino rock stars.
Oh, Rumino rock stars. Good.
But unfortunately, we're not talking about ruminants, but coming from Ruminococcus is one of those important fiber-degrading bacteria
that would
as
be,
well.
if we were doing a ruminant nutrition podcast, that would work perfectly.
Be all over that.
Okay, if you could ride any horse, past or present, which horse would it be?
This one.
Oh, you have to tell me what its name. What is its name?
This is My Little Pony.
Oh, my.
This is Rainbow Dash.
Oh, Rainbow Dash.
And I would want to ride Rainbow Dash, right? Because First of all, she's got wings.
So you can't see it because you're listening to us, but Belinda's got a decent sized rainbow dash in her hands at the moment, a plush one, and she's very cute, and she does have wings and they're sparkly.
And because she flies the quantal colours, that's why.
Oh, there you go. She's got a rainbow tail, the quantal colours. So Belinda's company is Quantal Biosciences, and I will put the link to it in the show notes, and also the link to remind me what the microbiome testing EquiGI. I'll put the link to that.
Microbiome testing service,
Yeah, I'll put the link to that in the show notes as well. And if you had 2 minutes with every horse owner in the world, what would you tell them?
Look, I think their microbiome of their horse is incredibly important. Around 70%, sometimes more, sometimes a little less, but around 70% of the energy that powers your horse comes from the microbes that are in the hindgut. So it's way more important than our own nutrition, way more important than our own microbiome. So be really kind to your horse's microbiome. Feed it well.
Mm-hmm. Love it.
Give it good rest. Give it good care, because what you're really doing is giving the best to your horse by doing that.
Excellent advice, and I agree wholeheartedly. Thank you so much, Belinda. This has been a lot of fun, and literally we could talk for weeks on this and still not have covered everything that is interesting to talk about in the horse's hindgut microbiome. But thank you for giving us a really lovely overview and some nice, really interesting little bits and pieces of, I guess, real front-running science on it as well.
Thanks, Nerida. It was an absolute pleasure as always.
Gosh, I love that episode. I remember way back when I was studying ruminant nutrition during my undergrad degree, we were taught that when we were feeding ruminants, we were essentially feeding the microbes, and the microbes were feeding the animal. Now, I'd not extended that concept out to horse nutrition for many years, but I think we can now all admit that our horses' microbes really are running the show. And the way we feed them matters a lot, because it will affect our horse's health, performance, behaviour, and a whole bunch of other stuff. And that brings us to the end of our podcast for 2025. It really has been fun getting this podcast up and running, and I want to say the biggest thanks to you for being here and listening. We have so many great practical, science-backed conversations in the pipeline for 2026, and we'd love you to subscribe so you don't miss any of them. And if you've got a friend who's deep in the horse nutrition rabbit hole with you, we'd love you to send them this episode. Happy 2025, and we will see you next year.